So let's go on to the next section. Isotope variations exist at subcellular levels and reflect secondary fraction events. And I want to tell you a rule in physics that you must remember. Conservation of mass. On our planet, we are neither creating nor destroying stable isotopes. So if you accept that, then life becomes pretty easy. And it becomes very easy because you will find in the literature conflicting pieces of information. Some people will say in the literature, particularly in the plant literature, that there are lots of fractionation events associated with respiration. Why? Because when they look at the CO2 that's coming off a plant or a cell, there are variations in that isotope ratio. Because there's variations in those isotope ratios, the statement is made in the literature that there's a fractionation event. Okay? And I would look at Kelsey and I'd say, well, Kelsey, tell me exactly what's being respired at the moment that that CO2 molecule came off. And she'd say, oh, I'm sure it's a carbohydrate. And Chris would say, no, I'm pretty sure it was a lipid. And Emily would say, really? I thought it was a protein. The challenge is to know exactly what's being respired at the time and whether or not it is a temporary difference because of some imbalance in metabolic reactions or whether there's a true variation in the fractionation. So I encourage you all, as you read the literature and as you express things yourself, unless you know the mechanism and you see what seems to be an isotope effect, use the word I have in the bottom right, apparent fractionation. Based on what I see, it appears that there might be a fractionation. Because Sean here is a real stickler. He works in biochemistry. He's not going to cut you any slack. He's going to say, are you going to tell me that alcohol dehydrogenase fractionates at 6 per mil on this day and 12 per mil the next day? Is that what you're claiming? Are you claiming that the physics of the reaction has changed? Well, if you use the word fractionation, you are making that claim. But it might be that the substrate which was feeding this dehydrogenase changed over time. And that was the reason that you saw the variation. So I always encourage you, if you're in your discussions of things and you don't want to put yourself out on a limb that's so thin that you saw yourself off and you really want to say that there's an effect, consider using the expression apparent fractionation. Now, one second, Chris. The Europeans have latched onto the variation in the isotope ratio of respiration, and they call it fractionation. In my lab 30 years ago, we used to do experiments, I'll come to you in a second, Chris, in which we would measure the CO2 that's come off a leaf overnight. And we'd get a value of minus 26 in the afternoon, or in the beginning of the night, and we might get a minus 35 in the middle of the night, and then it might go to a minus 20. We observed that as variations in the isotope ratio without knowing what was causing it. There's a tendency today to say, well, the bulk leaf value is minus 25, so anytime it's not minus 25, that's a fractionation. It doesn't need to be. Chris?
I was wondering if it's possible, or even if it's been done, I'm not familiar with either the literature or the methods, to look at fractionation in vivo with Rubisco and then separate some of the most common compounds in the leaf and look at how their composition.
That's what a biochemist would do.
And look at like the CO2 coming off a leaf and make inferences of which molecule or compound is most likely.
So a biochemist would pull this into a test tube and do that. There's a tendency to want to work with the whole cell, and you actually don't know if you have an intact cell which component is being analyzed or is being metabolized. So what I want to do next here is to establish, think of the word apparent, and recognize that if I sample the isotope ratio of bulk leaves, here I'm looking at tree height, so going from shade to the top, I'll get a variation. And I've alluded earlier that that variation is driven by a change in CICA. And if I look at the cellulose that I extract from that entire leaf, I see that it follows the same pattern, but it does not have the same values. Okay? I can do the same for branches of different species. And my point here is that when you're measuring the bulk isotope ratio, and the theories you've had have to do with the bulk carbon coming in, we must recognize that that 6-carbon sugar can be broken down into 3s and 2s, and those steps could have fractionation events. Okay? I could break things down into 2s, such as acetyl-CoA, and now have a carboxylation reaction, and those can result in variations in the isotope ratio within a plant. But unless there's a new law in physics that I'm unaware of, there is conservation of mass. And so that means that you don't destroy and you don't create new stable isotopes. So if you're going to say that there's a fractionation event, then you must be saying that in some pool I have these heavier light isotopes accumulating. The problem is that we have a literature that says that there are fractionation events, but they're very short term. They're just slight imbalances because we don't have any evidence of a pileup of carbon-13s or carbon-12s in a plant or in a system. So you need to take the bigger picture on this and recognize that if you now begin to look at the isotope ratios of different compounds, they can be different depending on how they were created, and that if you have a metabolic pathway which consumes all of the substrate, let me repeat this, if you have a metabolic pathway that consumes all of the substrate, there can be no fractionation. Okay? Even though Will here is going to say the fractionation factor is 6. The fractionation factor could be 6 per mil. But if you consume all the substrate, there's no fractionation effect. The only time you can have a fractionation event is if you have an open system and not all of the substrate is consumed. So everything I was talking about with photosynthesis today, we can have fractionation because not all the CO2 is consumed. But if you consume all the substrate, then you can't. I like to use the example of blue and red jelly beans. I love blue jelly beans. You give me a pound of blue and red jelly beans, I'm going to eat the first ones, the blue ones first. But since I like jelly beans, I'll eat them all the way to the very end. And so I might have discriminated early on, but by the end I've consumed them all and there's no discrimination effect. That's an important thing to keep in mind. Okay? Another thing to keep in mind is that if you break something down from, let's say, from a 6 to a 3 to a 2-carbon molecule, and now you begin to build it up, what you're building it up on depends on, it's going to be influenced by what other carbons are being added. So you need to know, you need to take that Merck chart. I mean, we used to keep the Merck chart up in the lab to say, oh, if we go through this pathway, where am I going to get a CO2 added? That's going to influence the isotope ratio. That's an important thing to keep in mind because you'll notice something very interesting. Lipids are isotopically light. Okay? If you're in my ecophysiology class, I'll tell you that the turnover rates of lipids are about 3 to 4 days. They turn over like crazy. Turnover of starches might be 3 to 5 days. The turnover of cellulose, doesn't really turn over. Turnover of proteins might have a longer time period. Because molecules are turning over at different rates and they have different isotope ratios, what's getting respired might reflect whether or not we are respiring lipids at night or sugars at night. It doesn't mean that there's a fractionation event. It doesn't mean that you can take this process and say, oh, respiration fractionates at 6 per mil. This is Jim trying to give you advice as you read the literature. Be careful as you read the plant literature. So there are clear differences in some of these patterns that are pretty interesting. Brett Tipple in my lab happens to be interested in a plant which is increasingly legal. And he wants to ask, are there biomarkers? And so what he does is he looks at a biomarker called a lipid, an alkane. I think these are C31s. No, C29s. So he's looking at a long chain component, which is part of the wax on the surface. And we measure the carbon isotope ratio. Lo and behold, it doesn't follow the bulk value. Values are always more negative. But notice that the slope is essentially 1-to-1. So there's a constant fractionation. Whatever the pathway is, it fractionates the same depending on whether you're growing the plant in the day, in the light or not. Now you might ask yourself, why is it that I have such a big variation in values? Well, that's simply because of those fractionation events. Now, you might ask yourself, if I'm looking at cannabis samples, and I see a bunch of values up to 25 to 30, where do you think those plants were grown? Outside. These are DEA seizure samples, the samples that were seized outside. Now the question is, how do I get a value of minus 40, minus 45, minus 50? I'm growing them downstairs in the greenhouse with my little methane heater. So, stands out. So what you need to think about as you're getting into this respiration business and looking at this respiration literature, ask, who could be a carbon substrate? Where do I find carbon-carbon bonds that have enough energy that I can use them for respiration? And what is the turnover rate there? And you'll see that we can classify plants as having high turnover rates and low turnover rates. Cellulose doesn't really turn over. And alkanes, waxes, it turns out once they're formed, they don't turn over. So the waxes that are on the leaf reflect the isotope ratio of the waxes when they are formed in the spring, even if you measure them in late summer. Nucleic acids, oh, there's a cool one. They don't change. Whether I'm looking at a microbe or a plant over the course of that cell, no change. But high turnover in carbohydrates, lipids, proteins, metabolites, and sugars. So be careful of what you're looking at as you make your claim with respect to respiration. So if the carbon isotope ratio of dark respiration varies over time, does it mean that fractionation, the fractionation factor for respiration is changing? I mention this because if you read a lot of the literature, people can say, yes, the fractionation factor in a young branch is different than the fractionation factor in a mature branch. And you should say, I trust your data, Maria, but now I'd like to challenge your interpretation. Have you considered this? Have you considered that? Okay. So let's explore this apparent fractionation with an example of what might happen. So let's take a glucose molecule that turns out, if we break the molecule apart and look at each of the carbons, the middle two are carbon-13 enriched. Let's not go over the mechanisms of why, but that is in fact the case. Has to do with how the molecule is synthesized. Let's say I take that glucose and I now break it into pyruvate. Why would I want to do that? Well, that's part of glycolysis. I'd like to have some ATP. So we break it down, get some ATP, and now we end up with pyruvate, which has 3 carbons, 2 of which have about the same isotope ratio, and the one in red or green, whatever that color is isotopically heavier. Now I'm going to take that pyruvate And with a pyruvate dehydrogenase, which is PDH, I'm now going to cleave off another carbon molecule to make it a 2. And it turns out the one it cleaves off is the heavy one. So the CO2 that comes out for respiration from the C3 and C4 positions has a value of minus 21, even though the bulk glucose molecule had a value of minus 25. You might ask yourself, how does it get to be so enriched? I'll tell you later if you want to know, but it happens through very predictable chemistry. Now, I've got my pyruvate dehydrogenase cleaving the pyruvate, and it gives me acetyl-CoA. And at this point here, a number of wonderful things happen. I can take that acetyl-CoA, and I can run it through the rest of the respiration cycle, the mitochondrial cycle. And then in that case, I'm going to respire all of the carbons. So if I integrate over the course of the night, I started off with a molecule at minus 25, and I end up with CO2 over the night accumulated. That's a value of minus 25. But if I now, instead of looking at over the entire night, look in the beginning, I might get respiration of CO2 that has a value of 21. And I'm going to run to the press and say, look at this, guys, there's a fractionation during respiration. Well, you do have a difference, but it's because you now have an accumulation of acetyl-CoA. The question is, what happens to that acetyl-CoA? I can break down that acetyl-CoA into CO2s, in which case I have another fractionation, but now in the opposite direction. But the integral will be no fractionation. Or I can do what happens in plants a lot. And that is that the acetyl-CoA now gets split. Some of it goes to mitochondrial respiration, and some of it goes into building new blocks. And because that molecule is isotopically light, those molecules that get built will be isotopically light. So lipids, terpenes, things that are built off this 2-carbon in other organelles end up being isotopically light. So you can see how you can, by going down to this 2-carbon and then now building up, how you can have a bulk value of maybe minus 25, but different components at different values. And it all depends on what you break things down into before it gets built back up. And fractionation generally is going to be a function of molecular weight. Okay? So when you get to the 1-carbons and to the 2-carbons and to some degree the 3-carbons, you can have fractionation events. The biggest fractionation events you're going to get are at the 2-carbon stage, because the only 1-carbon stage is photosynthesis, typically. Okay? Now, this turns out to be true for animal systems as well. So the rules I'm giving you are general rules. Okay.